So Sam Lipson serves as a Senior Director of Environmental Health for the Cambridge Public Health Department. He established the Environmental Health Division in 1998 and now oversees the enforcement of local and state regulations pertaining to hazardous chemicals, biological, and mineral exposures within the city of Cambridge. His responsibilities include environmental health policy, air quality research with academic partners, hazmat and indoor air site investigations, emergency preparedness and planning for major hazardous material events and anticipated climate change impacts. In addition, and I think of a particular interest to all of us in this audience, he coordinates the Cambridge Biosafety Committee, which is a citizen panel authorized to permit and inspect all life science labs. Sam's talk will focus on how to achieve public safety and health. Sam?
Thank you, Steve. I am the Senior Director of Environmental Health. I think Steve or Goody gave me a great intro, so I'm not going to spend any more time on this. I've been working for public health in Cambridge for about 25 years. I, by the way, consider myself the Bay Area is sort of a second home for me. I went to Berkeley as an undergraduate. I lived out there for about 8 years, really miss it a lot. So here to talk as a traveler, as a visitor from not only another place, because Dennis and I are coming from all the way from the East Coast, but also kind of another time, because the events that led up to us having the kind of oversight we have in Cambridge really were set in motion in 1976. And there was a lot of consternation and anxiety and uncertainty about some of the new work going on with restriction enzyme, with the ability to introduce genetic material from one microorganism into another. And this breakthrough caused concern, not so much in the public initially, but among other biologists who were frankly uncertain about what the consequences might be and didn't feel that those uncertainties were being addressed correctly. Eventually, the level of concern grew to the point that they did not feel that they were being addressed through the university. That's really both at Harvard and MIT. And they came to the Cambridge City Council in 1976, which ensued a series of discussions between biologists at both universities and council members, which are quite entertaining and kind of demonstrate how difficult it is to talk about these topics. But I want to give you a little bit of background on sort of what the moment was like at that point, because as for those of us of my generation and older, they can certainly recall that the 70s were a period of time when a lot of distrust towards government institutions, military, even academia, was on the ascendancy. Information about nefarious activities that had been conducted by the government had become known at that point, and there was skepticism, a great deal of it. The initial introduction of this new area of biology through the work of individuals at MIT, Harvard, Stanford played a big role as well, introduced a series of uncertain risks that were very hard to talk about because it wasn't fully understood what the consequences would be, even unintentionally, of doing that work. This was acknowledged and recognized by the scientific community, by the biologists, and it was, I think, a really strong example of taking an issue which was hard to define and bringing together individuals who had great knowledge about it and beginning to build a roadmap and a set of rules. It's almost like teaching you and fellow scientists how to drive in a new environment. There was an early conference in '73 on oncogenic viruses. There was a couple of other conferences in the '73 and '74, but these led up in many ways and supported the work of what we call a CELMAR 2, which occurred down in Monterey in 1975, which is really where the hard work of bringing in biologists who were familiar with genetic manipulation at that time, the tools that were available, virologists, other infectious disease specialists came together, folks from NIH, from various universities, public and private, and they began to basically hammer out a set of rules, and that's eventually turned into the NIH guidelines, which were finally published later in 1976. When NIH finally did publish their guidelines, there was indeed a roadmap that the city of Cambridge now had the ability to look at. Now, the concerns in Cambridge were brought to the attention of the council before these NIH guidelines were promulgated. But as soon as they were put in place, there was sort of a coordinated effort on the part of City Council to create rules that tied directly to those guidelines. And that's what happened. So I'm going to skip forward a little bit. We have a lot of material. This is interesting historically. There's a couple of videos at the end of my talk that you can watch to get a little bit of background, not all of this, but much of it, about the conversations and conflicts that occurred at that time. I did want to speak as a public health official as well for a moment. The introduction that we got to the biosafety levels versus risk groups was excellent. It's just of a quality that we don't normally get in public presentations, and I hope you can all appreciate that. But these are a number of factors that are taken into account, some of which were discussed, I believe, most of which perhaps, during the introduction by the biosafety specialists who were on before me. Obviously, consideration of the risk group is critical. Risk group alone does not tell you what containment level is appropriate, because it could be mitigated in many ways, not only by how it's used, and uses was discussed as a final determining factor in the biosafety level, but also in whether or not it's been altered so that it's no longer capable of infecting a person or anything. If it can't reproduce itself, then it can't really infect. So replication and competence is another factor. For any given pathogen, the dose at which it can successfully infect a person is a driving factor of how much risk is involved. The virulence, if infected, how serious is the illness that ensues? Whether that there are any chemical toxicants or biological toxins involved in the procedures that are being carried out with this pathogen. Some organisms naturally produce biotoxins. Environmental stability, if there was a release within the lab or within the environment in general, how quickly under environmental conditions or normal conditions would it become deactivated and break down? Some organisms don't last outside the human body for more than a few minutes, maybe less. Are the procedures involved in that work potentially producing aerosol? Or are they involving procedures where no aerosol generation is likely? The product produced by genes that are being inserted into some of the host cells that are being worked on. Early on, there was a great deal of concern that there might be introducing gene functions that would increase risk. That has not largely played out. That's one of the things we might call gain of function, but it does happen in some areas, and it's still an area of concern. What is the range of organisms that might get sick as a result of exposure to that organism? Is it just humans? Is it other animals? Is it not humans? Only other animals? Route and ease of transmission. We learned a lot about this during COVID. We have vaccine availability and safety, and that's more about population protection. If there were a release or something that was transmitted to a larger population. The efficacy of therapies and treatments that are available. How long are symptoms that a lab worker might get as a result of an exposure delayed before they might have an indication that they themselves are infectious. That latency can create opportunities of risk because they may be then traveling and shopping in the community. Disease surveillance systems. How well set up is the hospital and public health system in that area to pick up on an illness which would be unusual or unexpected? And then the overall public health infrastructure, hospitals and public health departments. So I'm going to be mostly here to pitch the idea that local regulations have been a very big success in Cambridge, and it has brought a lot of really good trust and practices. So this is the thrust of what I'm going to talk about mostly. Increasing internal commitment to worker safety and risk awareness because of the attention being paid to the internal safety committees is something that we have gotten feedback from people within companies is absolutely a factor. They know the city is looking. They know that they're expected to share minutes from these meetings, and there's a kind of an upward pressure on better practices. Companies get more predictable regulatory oversight and credibility because they're really sharing a lot of pretty critical information about what they're doing with the public health department. And they're also getting a really clear set of rules that if they follow, they have a right to operate. So both their willingness to be transparent and their right to operate are benefits going in both directions, I would say. I have a link here that is to MassBio. Dennis mentioned that this is the big lobby group for the biotech sector in Massachusetts. They have adopted a position and have for quite a while that they want to see communities adopt local regulations based on the NIH guidelines. And they even have a grading system for towns as to how ready they are for biotech. And I think this is a really strong reflection that our approach to oversight has not been onerous, and it has brought a great deal of trust. And when there's skepticism in the community at large, this has been a very powerful tool. And that the industry doesn't have a problem with it. In fact, they see benefit. So some other benefits I think I mentioned already: gaining public confidence through transparency. Another element of the program that we run in Cambridge, which I think works in communities that have a lot of labs, is to actually have public participation mandated and work with those community members to train them to sit on committees. Some people have a great science background, they might even be retired from the field that they're reviewing, but many people don't. So we do spend a lot of time working with community members to try to make sure that they have what they need. We do trainings periodically. In fact, we're probably going to do one later in the spring. So I would mention that the recent build-outs in Cambridge have become an issue, but as Dennis pointed out, it's mostly because of noise and light pollution and overall density and those issues. I feel great pride in the fact that a very skeptical city of very sharp people feel that there are issues, but the issues are not whether they are being put at risk by the labs because of biological risk. And I think that is the result of a lot of the work we've done in the city over a long time, about 45 years. So I'm going to talk a little bit about how people generally have seen risk. This is a more general observation. Almost anybody could make these observations, I think. But from those early days in the mid-70s, when there was a great deal of concern and opposition to the work involving recombinant biology, later synthetic as well, that routine work and the fact that they're using such an unusual, now not so unusual platform of tools, of genetic engineering tools, is no longer so exotic and not the sorts of enormous concern in the community in Cambridge for sure. And by and large, until pretty recently, I think that the typical biotech work has not elicited an enormous concern. But that's mostly low risk work, BL1 and BL2. There has been a more recent wave of information being pushed out, some of which I've gotten to see through social media, that's basically disinformation, just describing secret government labs doing work that is not remotely of the sort that they would describe, and often pointing at buildings where they're doing very routine BL1 and BL2 work and implying that somehow it's part of a larger government system that's not being shared with people. But indeed, the opening things up and bringing sunshine into the work that is going on is really the best approach. In Cambridge, at least, and by and large, I think in the Boston area, I think you would say that bringing in, yes, increase in housing prices, a lot of highly paid jobs, high skilled jobs have had an impact on our local economy. But as far as an industrial sector, it's been pretty low impact. It's largely about parking and nuisance, the kinds of nuisance that you would get with large office buildings as well. But I will say, and I think we're here today to talk a little bit about higher risks, not only those that are already known and understood, because people know that there are many risks out there from hazardous waste sites and from chemical manufacturers and even from some very high-end biological research facilities. People continue to have worries about them, and they should. Within the biological community and the academic and government labs, I would say that there's been an ongoing concern about research that could be used for nefarious purposes, not the safety of the work itself, but how the work could lead to unintended consequences as a result. They call that dual use research of concern. So thinking ahead about how that work, sort of life cycle of that knowledge, where is it going? Emerging pandemics have brought a lot of attention to gain of function studies, which have been a really important tool, but perhaps a new era of skepticism or scrutiny will come to certain gain of function studies as a result of concerns that risks are being generated in labs. Whether that happens to have anything to do with COVID or not, we don't know at this time. And then of course the build out after 9/11, there was a huge amount of money pushed out by the federal government to fund BL3 and BL4 labs to get built out across the country. The BL4 labs especially have brought a great deal of anxiety and scrutiny. So this issue is not put to rest, but this is an effort to distinguish sort of the typical low-risk biotech typical work from some of the specialty labs that are out there that have higher risk work going on. And the public has shifted focus also to the applications of genetic technology rather than the exotic nature of the fact that we can do it at all. And this has come out in terms of concern over genetically modified foods. So patent control over seed stock, monoculture seed stock, where you have a genetic component that has been patented is an enormous concern. We saw how people reacted to the idea that an mRNA vaccine was now being used for the first time. And that raised in many people quite a lot of anxiety and still does. The overall concerns about the biopharmaceutical industry are certainly include things like efficacy, safety, cost, fairness, and whether or not they're doing relevant work that people really need to have done. There's other concerns, of course, about gene therapy, and there are reported adverse events in gene therapy. So it's not at all out of the realm of concern of people who might have to consider whether that's appropriate. And then I don't know if everybody here picked up on the news cycles around CRISPR, but that is an ongoing example of powerful new tools keep coming and are available to scientists, and the consequences could be that they're used in ways that not everybody thinks is ethically appropriate. So back to the overall intent of our oversight program in Cambridge, it's really tied to the culture of accountability and the culture of safety. And by making sure it's really clear to labs that are coming in that they, in fact, have an obligation to be transparent, and people in labs moving into Cambridge know that, there's really very little drama. We work with them on how to protect proprietary information. There are many ways to do that. We've brought an emphasis on the high quality biological risk assessment as part of the work of the IBC that should in fact be documented. There's a lot of discretion. IBCs should be granted and are granted, but they should explain through the minutes and through the meeting why they've chosen to assign a certain containment level to this protocol or that protocol. The biotech tools and source materials are changing all the time. I mentioned CRISPR. This is not a static set of variables. The idea of having an ongoing biosafety committee in Cambridge has been useful because we have to continually update our knowledge so we understand what's going on in these labs as well. Transparency, strong safety culture contributing to public assurance. This just comes up a lot. It comes up with individuals I talk to, when I engage with the media, other public elected officials, the fact that they know that we're getting good detail that's appropriate level, really, to understand the nature of the work going on in these labs goes a very long way. We have some emerging challenges in the work we do as well. Incubators are these laboratories for researchers who are in very, very small groups or individuals. And Science Hotel's a similar approach where you have a kind of a turnkey laboratory where you can kind of drop in and be an independent researcher because you don't yet have all the funding to to produce your own lab, to build it out. Maybe you haven't shown efficacy of your principles. And so these Science Hotels and incubators are popping up around Cambridge, and I'm sure in the Bay Area too. But they're also raising some difficult governance questions. Who's coming in and out of those labs? What work are they doing? How is it screened? What is their accountability to the city? Complex lab ecosystem out there includes a lot of contract research organizations that do, for instance, a lot of animal work, not done in the laboratory that we regulate, but being done entirely off-site. There are also a lot of change in the biotech sector, so it can be difficult to keep up a little bit because mergers and acquisitions, name changes, very high staff turnover. These are ongoing challenges for me. And biotech sector expand to areas with limited oversight or transparency, that is physically, geographically. If they're in Cambridge, they're getting a lot of oversight. If they move to, say, a town like Woburn, Mass, outside of Boston, there are no rules at all. Other than the non-biological oversight they get through OSHA and through the state, they don't really have a lot of obligation to report anything. We did develop our biosafety regulations here and have changed them a little bit over time. But I think it's what I would like to impart to you in the Bay Area and those who are listening is that there's some really important questions to consider ahead of time before you just dive straight in. And a few of these include whether or not you're going to have health regulations. Is this going to be a role for the health department? I think that it is a good role for the health department. But if you really want to look at something that's strictly limited to zoning and land use, that isn't obviously within the purview of the health department. So that's a question. So another is, what is the biosafety level that would be allowed? Would BL1 and BL2 be allowed without any, without anything more than kind of a standard permit type oversight system? If BSL3 would be allowed, what would be required additionally for that? Would the community or county that would consider regulations want to take on the additional work of having a biosafety committee, a public biosafety committee? If there are enough labs and enough concern, that may be appropriate. If you have less public participation in the overall process, like having public meetings, having to train individuals to sit on IBCs within companies, it's going to be a lower burden with the health department. But there may also be less trust that comes with that. So, finding the range of how much effort and involvement you want to engineer into the oversight is important. Determining whether regulations will include animal care facilities or just microbiological work is really important. Cambridge has a separate lab animal ordinance. We have a commissioner of lab animals, but that we also have such a large number of facilities that it probably doesn't make sense in a lot of places to do that. One approach that I thought about a lot for communities that maybe if we had done it differently in the beginning is something I call a hybrid approach, where you only register BSL1 labs, you permit BL2 in the way that we do in Cambridge, and then BSL3 has some additional requirements, as we do as well. And they may be land use requirements or coordinated long-term planning or zoning requirements as well. As I said, local public health has really been the anchor for biosafety oversight in Cambridge and in many other communities around Boston. And I think they really bring a level of credibility to the question that isn't brought by other departments. So I know that I've gone fairly long, and I do want to get through this material, but I don't want to go so far over to not give enough time for other people. So these are responsibilities of a permit holder in Cambridge, and in typical of the regulations that have spawned outside of Cambridge, but based on our regulation in the Eastern Massachusetts. Compliance with NIH guidelines. Often the MBL is cited as well. IBC per NIH guidelines, it should include two community members. Different communities have different restrictions on whether they have to live in that town or not. Appoint a biosafety officer. Ensure IBC members have the right background in the material being reviewed during the meeting. So if you have a larger company, you might select members of your IBC, internal members, appropriate to their area of specialty. Make sure that those IBC members, or even in-house members, are properly trained, but certainly offer additional background material and training to the community members as well. Establishing a medical surveillance program, post-exposure protocol. Again, if you're dealing with infectious agents, this is especially important. A standard med surveillance program that we look at, we require them anyway, are pretty modest because they're not really dealing with infectious agents in most cases. Report potentially infectious exposures, bites, punctures, and cuts. They have a little bit of time to report, but that is a requirement of the ordinance and the permit. Renewal with the IBC documentation for the previous year. In other words, show us that you met, the IBC met, that the minutes are sufficiently detailed and captured a discussion, a conversation, that protocol summaries are also part of the documentation requirement if they were newly reviewed, or even if they were reviewed and renewed from a prior period. If they were addressed by the IBC, they should be summarized and included. And then the membership, the roster of the IBC. We also require that new permit applicants come and present directly to the Cambridge Biosafety Committee. These are public meetings. Anybody can listen in. They've been done over Zoom recently, but it's a bit of an ordeal, but a ritual. And we make it easy for them, give them a template for the talk and everything. This presentation I mentioned, that the template that we give them includes purpose of research, overview of vectors and agents and procedures, floor plans, what areas are assigned what containment level, waste removal pathways, what are points of access, talked about med surveillance agreements. We permit compliance with other permits, state, local and state, service contracts, who have you identified? Somebody you've already identified or hired somebody to work on removing bio waste, chem waste, pest control, air balance, other equipment certifications in the lab. Summary of safety practices, training, lab policy should be put into a comprehensive biosafety manual, which we review before the permit is granted. And the resumes for all the members of the community who serve as outside members are shared with us as well. Lab inspections are a requirement, and I do all of them. When we have BL3 labs, we bring the entire committee, but for most of them, I go through by myself. Lab conditions overall, flow of consumable supplies. These are about how well the lab is organized and run, how much clutter is there, how well they thought through stuff. That's a lot of what I'm looking for. Pathways of removal of chemical waste and biological waste. Where is it being stored before it's picked up by a vendor? Wastewater treatment and pH neutralization. Lab signage, is it appropriate? Do they give sufficient details? You're going into lab areas with higher risk or agents with higher risk, where agents with higher risk are being worked with. Certification of the equipment in the lab, BSCs and hoods. And then general ergonomics, we talked about material flows, noise, safety. And I always save time at the end of the inspection to talk to them about the IBC and what we want that IBC to look like in terms of its role. The IBC should be thought of as at the center of the biosafety program for that company or that institution. They're not just there to approve protocols. They're there to think and talk about and discuss policies and incidents that have occurred. And so we talk a lot about that. Virtual inspections are now being used for very minor amendments at sites that have already been inspected. Further notes on BSL3. This is my next to last slide. They do represent a large increase in potential risk. Now, we don't have very many in Cambridge anymore. We had, I think, up to 5 at one time. We're down to 3 now. Boston has a lot more because they're much more tied to the medical district. We have a cluster of medical schools and hospitals in Boston that have affiliated BL3 labs for related work. It's much more likely to contain it almost certainly will contain live infectious pathogens that are replication competent. They can infect somebody. This is so different from most of the work at BL2. Engineering standards are very high, and I'm so glad to see the breakdown of what some of those standards were in the previous talk. But validation is and needs to be completed by a third-party commissioning agent, and there are a number of firms that just do this. They do BL3 and BL4 commissioning. The emphasis on safety trainings and protocols and risk reviews is really important. So again, even if you don't have an overall regulation for other biosafety containment levels in the Bay Area and some of those counties, figuring out how to act fairly soon to recognize that BL3 labs require a level of some level of scrutiny that other labs may not really require. It's important to do that quickly, I think. Emphasis on also additional costs to retain safety consultants. Again, even if you don't do a full soup to nuts biosafety oversight program like we have, if you're going to try to regulate BSL3, you should really include the costs associated with bringing in consultants, because this, both for inspections and for reviewing documentation and the commissioning documents associated with that facility, really requires people with that kind of background, and it costs money to do it. And I think that even without, so I think I've covered this slide, and I have a final summary recommendations. I see Steve there because he's been so tolerant giving me extra time here. I appreciate it. But I would say most life sciences labs in the biotech sector and the private sector are using non-infectious agents. They're replication incompetent, not entirely, but from my experience with we have about 180 companies in Cambridge. So I have a pretty wide array of experience with that. Some research requires live agent, most of it's pretty low risk live agent work. Again, BL2 mostly. Those labs, however, that are not getting any federal money to do research, if they're doing BSL3 work and they are a private lab, they are not subject to any of the NIH guidelines that we've talked about, and that will come up, I think, in a later talk. You've heard my pitch about local biosafety regulations. I feel very strongly about that. Local city and county government needs to consider whether lab animals is something they want to include in that regulation or consider separately or regulate not at all. But that's going to come up in the discussions that occur with you in public hearings, I'm sure. And it needs to be addressed now, because BL3 labs are going to be built if there are rules in place. And I think some rapid action plan even in lieu of a longer, larger biosafety regulation approach, is really warranted for BL3 labs. I hope you appreciated this, and I really want to let you know that it works. It works in Cambridge. It's not easy, but it really helps bring a lot of value, a lot of trust, a lot of public knowledge about what's going on in the sector. So I want to make sure you know that these 2 videos are linked at the end. You'll get a copy of this if you want the presentation, and you can go check them out. Thanks very much. Steve?
Wow, man, that's so much. Thank you, Sam. That's awesome. Yeah, thanks for everything that you do for your community, your region, and as well for coming to present to us here on the West Coast and sharing your obviously deep knowledge with us. Your talk was both informative and insightful, and we're very grateful. One thing that's obvious is that we on the West Coast have a lot of catching up to do, and we need to get right on the ball, I think, to start addressing these things sooner rather than later, and whatever model is adopted.